My name is Barney Balch. I'm a senior research scientist here at Bigelow Laboratory. And I'm going to be talking to you tonight about a talk called Distant But Connected, a journey to discover how Antarctic algae shape the northern oceans. But I'd first like to welcome you all to this Café Scientifique. Bigelow Laboratory is a nonprofit basic research institution focused on the lower end of the marine food web. And the screen just went blank. All right, I'm not sure if people can hear us. Yep, we're still live, okay. We just lost power here at the lab. So Bigelow Laboratory is a nonprofit basic research institute focused on the lower end of the marine food web. Our mission is to study the foundation of global ocean health and use our discoveries to improve the future for all life on the planet. Our vision is a world where scientific understanding of the ocean turns the tide on global challenges and unlocks the full potential of the ocean to support our society. My personal connection to Bigelow is that I grew up with this laboratory. I've worked at the laboratory since its founding in 1974 as a high school student. And from there, I went on to do my undergraduate work at Cornell, and then I did my graduate work at the Scripps Institution of Oceanography. From there, I went and taught at the University of Miami Rosenstiel School for 8 years. And what gets me up in the morning is that Bigelow is unique as a place to perform basic research about the world ocean. It's a place where by the end of each day, you can walk out knowing that you've learned something new about the ocean. And as a nonprofit research institute, the discoveries, solutions, and inspiration we create are made possible with the help of our generous supporters. And I want to start today by thanking each of our donors, board members, and advisory board members for their support. I'd also like to extend a special thanks to H.M. Payson, a Maine-based financial advisor that is again sponsoring this series of talks. You're going to see a Q&A button, and which you can use to send in your questions at any point during the talk. We'll take a brief pause partway through for you to submit questions, and then there'll be some moments at the end for you to also ask questions. Please use that question and answer button, and I'll try to answer as best I can. You'll also notice a raise hand and a chat button, but these features are not available for this event. So this talk is about two things. The first part of the talk is about the process of doing field oceanography, and I call this the journey. And the second part of the talk is about a gigantic ocean problem, how Antarctic algae shape the northern oceans. So the journey, and this is not so much about the science, but the how we get from an idea to actually get out to sea and test some hypotheses. The field science involves a lot of logistics, especially when you go to sea. The planning time for cruises like the one I'm going to describe for you is comparable to going into outer space. I'm going to show you a journey timeline here. It's not meant to be a downer, but it is the reality of studying the ocean. There are just so many logistics that one has to deal with. And the other part of the difficult aspect of this is Mother Nature doesn't reveal her secrets easily. The end result is well worth the investment if we can learn how the ocean works. But the mantra that one has to say to oneself as you go through this is go with the flow, and you'll see why I say this. So the timeline for the science I'm going to describe for you today actually started with an idea in 2015. And I did what I usually do when I have what I think is a good idea, is I write a proposal. And this proposal went to the National Science Foundation. This is the first proposal that we submitted, went in mid-2015. And the earlier part of 2016, proposal number 1 had been rejected, which is a real bummer. It had been reviewed anonymously by 8 peer reviewers, and then it went before a panel of 15 anonymous scientists. And actually, we got pretty good reviews, but the scientists that reviewed it said, gee, we think you should add a few more people, add another cruise, things that I was reluctant to do because it would make it more expensive. But we went ahead and rewrote the proposal. And because of the rules of the agency, you have to wait a year before you can submit this idea again. So we wrote proposal number 2 in the beginning part of 2017. And then mid-2017, we found the proposal was actually funded. So we're about 2 years into this, and we found that they liked it this time. So in that proposal, we had a cruise that we were proposing to do in January 2018. We wanted to get right out to sea. And so there on the timeline, you see that original cruise, we'll call it 1A. And then we discovered that there weren't going to be any ships available in the beginning of 2018 down in the southern part of the Indian Ocean, which is where we wanted to be. So they said, you're going to have to propose to go in 2019 instead. So we started planning for cruise 1B in May of 2018. And we shipped all our gear to South Africa. There were 3 containers full of instrumentation, and you name it, we shipped it over there. All of this was going to Durban, South Africa. And at the very end of the month, we found out that cruise 1B had been canceled. The ship was not going to be able to go to sea. They had a major engine malfunction. They had only left their midlife refit a year before that, and they had some major issues that came up. They were stuck in the shipyard for the next month. So we had to ship all the gear back from Durban, South Africa, back to the US. And we started then planning cruise 1C, which had been put off now till January 2020. And we shipped the gear to South Africa dutifully, and all the chemicals and all the containers. And we actually made it to Cape Town, South Africa in January. It was about, we flew at the 19th of January of this year. And we were, as you can imagine, very excited, almost 5 years from the original idea. So this is the research vessel Thomas Thompson, and it sails out of Seattle, Washington. It's 276 feet long. It's a global class oceanographic vessel. The US has 3 such global class vessels in their fleet. And the Bigelow crew here that was aboard, this is when we had first arrived at the ship. Sidney Greenlee was a Colby College student. Colin Fisher, this is from left to right. Colin Fisher, a technician in our lab. Dave Drapeau, the one with the hat on. Bruce Bowler in back with a white shirt. And then to his right, Sarah Rauschenberg, who was working with the trace metal group on the ship. Myself, and then we had a school teacher, Marcy Train from Long Island, Maine, who was helping us do what we call broader impacts to translate our science to school students. So when we got to this ship, which was in Cape Town, South Africa, we arrived to find a norovirus outbreak, which had happened aboard the ship on the previous cruise to ours. It was a cruise of undergraduate students, and as happens on ships when these things get out of control, probably some 65% of the crew, the 50 people on board, were affected by this norovirus. And so when we arrived, we were met by people spraying Clorox solution on the walls and on the floor, and basically trying to clean up the ship. And that they did. They did it very well, because none of us came down with it after the ship left, thankfully. And 2 days later, after our arrival in Cape Town, we came across 2 stowaways. As part of this interesting journey, the marine technician on the ship, she was up forward in the stowage area for personal flotation devices and survival suits, and she was up basically up high on a shelf that went up into the forepeak of the ship. She had a headlamp on, and she came face to face with a stowaway, and she carefully backed out, and as it turned out, she unknowingly had crawled over another one of the stowaways who was buried in the life jackets up front. And so this, needless to say, this is a first for me for all these years going to sea. This is the first time we've had stowaways on the ship. They were ushered off the ship. They actually tried to break into the ship again at about 3 in the morning, and they were met by folks at the gangway. But you know, this stuff happens, and you just have to say, go with the flow. The problem is if you get out to sea with stowaways and they appear, then you've got to turn around and go back, and it costs a lot of ship time. So we departed on the 25th of January, 2020. And before we departed, you see some of the dogs in the picture. They brought dogs on to try to find any other stowaways. And then once they cleared the ship, we were able to go. So we're off after 5 years of planning this program. That's Cape Town in the distance, and we're headed out to sea. And this is where we were going. We were planning to go, that dashed yellow line. And that dashed yellow line basically was to take us 38 days. And we were going to head out basically down across this region here, down to some areas around here where we were going to work for a couple of weeks. We're going to head all the way to 60 degrees south and come back up, survey the areas that we had surveyed before, and then head north up to about 30 degrees south, and then head for the island of Mauritius, which is in the southern Indian Ocean. So we got 2 days out of Cape Town, and it was about 2 a.m. in the morning, and this is what happened. The lights went out, and the lights went out, everything stopped. And we all said, uh-oh, there's no propulsion. It was like being on a ghost ship. And you could hear the water sloshing. And there was a coupler between the main engine and the main generator that broke. It can't be repaired at sea. Uh-oh. 5 years it took to get to this place. We're going to have to stay near Durban, South Africa for the rest of the trip. So the captain and I conferred. Basically, we had, at that point, 1 main engine, so there was no redundancy, and an emergency generator. And we could still make way with that just fine, but if something happened to that other engine, then it was a much more difficult situation. So we opted to stay closer to Durban so that we could duck in as soon as the part arrived. So now this is the cruise track. I basically, that part that was all in yellow, we had to abort that part of the trip, and everything we did, we stayed right around here. And then, without telling you about the science yet, just part of this journey, we returned to the island of Mauritius 38 days later, and we had gotten some great science done. And of course, we did have to duck into Durban, and we lost some precious time during that. It was almost a week that we lost out of that trip. But we returned to the island of Mauritius to find the novel coronavirus, which had reared its ugly head. This is in the beginning of March, and all the flights were getting canceled. The agencies like the National Science Foundation were being closed, and there were lab closures, and it was a pretty extraordinary re-entry into civilization. So that's just a little bit about the journey that was involved in this trip. Not really about the science yet, but I'm going to pause here for questions about the journey before I go on.
Thanks, Barney. We do have a few questions here. The first one was just how typical is this timeline for ocean research?
This is probably a little slower than some, but you know, it is not uncommon to write a proposal and have it returned. I mean, that's have it declined, and so you have to anticipate that. But you know, field research, it does take time, and these ships, these are major research vessels. To get them to a certain part of the world, they're not. You can't just ask them to be in a certain place in January of 2018, because that depends on where they were in November of 2018. And so, so they basically play this game as they learn about the cruises that are happening next year. They try to adjust the assets to the area that they're going to start work in. But when a proposal gets funded like ours did, mid-year, they can't get assets fast enough to that particular area. There's so much demand for the cruises.
Great, thanks, Barney. We have a couple people asking how many other groups of scientists were on the ship with you.
Yeah, so I wrote this proposal with, there were 5 institutions involved. Bigelow Laboratory, we were the lead on the project. Florida State University, Peter Morton, he provided the trace metal expertise for understanding trace metal concentrations. That's trace metals are these like iron, which is very important for the growth of algae in the Southern Ocean, typically. We had a group from Woods Hole Oceanographic, Dennis McGillicuddy, and they were operating a device called the Phytoplankton recorder you'll hear about. We had a gentleman named Nicholas Bates from the Bermuda Institute of Ocean Science, and another modeler by the name of Matt Long from an institution called NCAR in Colorado. And then we had foreign participation by a group, Colin Brownlee from the Plymouth Marine Biological Laboratory in the UK. So it was those groups that were involved.
Next question is about the changes that were made to the cruise track, and just wondering who has the kind of authority to make that decision, and did the main laboratory need to be involved in making those changes?
Yeah. Oh, that's a great question. No, basically the captain conferred with me as the chief scientist to say, look, this is what happened, and here's what we need to do. He said, you know, I'm reasonably comfortable going with one main engine, and then he threw the ball into my court and said, can you guys do the science that you need to do in order to solve your scientific questions by staying safely within range of Durban, South Africa. And that caused me, I had to spend a bit of time thinking about that one. And as you'll hear later on, there are certain reasons we chose the sites we did. And then I ran it by the captain. Ultimately, he has the authority. And then we tell the agencies, basically, this is what happened. and that's just the way it is. And so when the part arrived in Durban, South Africa, we planned it such that we had achieved one of our goals, and then we could dodge in to Durban, get the part installed. They actually literally had to separate the engine from the blocks that held it down, which is why you can't do it on a rolling ship, and push this thing back to put in the new coupler. And 8 days later, we were back out at sea again and working on the second objective, knowing that we had actually aborted that meridional transect. So.
Great. We've got a bunch more questions, but we'll just do one more for now.
Okay.
And then maybe we can cover some more of these at the end. But someone's asking, just saying that your costs must have gone up a lot as you ran into these issues and potentially required you to exceed the funding of the original grant. And just wondering, does the National Science Foundation provide funds in a case like that? And were they able to help out with some of the issues you ran into?
Yeah, the answer is thankfully yes. Yeah, we lost a lot of time. And of course, I've got the salaries of the people that work in the lab and that were basically trying to run the operation and planning and packing and shipping. And so, yes, I had to apply to the National Science Foundation for a supplement and justify it based on how much time we had lost. And then we had to wait and see if we were going to get that supplement. And thankfully, actually just yesterday, I received the supplement for the next cruise, which will allow us to do it. So thank you, National Science Foundation.
Okay, we'll hold the rest of them for later, Barney.
Okay, all right. So now I'm going to tell you a bit about the science, how Antarctic algae shape northern oceans. And to follow this problem, you have to think really big. And this goes for actually many of the ocean problems that we deal with at Bigelow Laboratory. There are large-scale features that are incredibly important to how the ocean works. The importance of these large space and time processes that dominate in the sea. So to give you an example, I'm going to go to an example of weather. And here you see a map of the Booth Bay region in mid-coast Maine, and I superimpose some temperatures there. And you see there's some pretty warm temperatures up the peninsula, and you're looking at the local situation, but you might not really realize what's driving those until you pan back and you look at something like the jet stream. And the jet stream, which carries weather, carries moisture, and the position of that jet stream actually dominates the local influences in weather around here. And so if you really want to understand meteorological problems, you need to be looking at these large processes. Another example, I'm going to use the same map of the mid-coast Maine, but now I put some Celsius temperatures here in the water. And if you're looking at warm temperatures, which would be typical of a summertime, summer temperatures around these parts, you get offshore, it's a little bit warmer, but you might not realize, if you tried to look at those data, you might not realize that these waters are buried within a much bigger pattern. This is a map showing temperature anomalies from the North Atlantic, and the redder that water is, the warmer it is. This is a figure from Andy Pershing. And so the reason you might be seeing warm waters in the mid-coast is because you're buried within this gigantic pattern of warm water, which is being driven by a very big process, whatever that may be. So when studying the cause of things, think big. Well, even phytoplankton variability, the microscopic algae of the sea, they also, their variability is driven by big patterns. Let's see if I can get this video to start. There we go. Okay, so these are, you're looking at remotely sensed, so satellite detected, ocean color measurements that show you the amount of algae in the water. The green areas are areas that are high in algae, and the blue areas are areas that are low in concentrations of algae. And you can see some large patterns like that big tongue of enhanced productivity across the equatorial Pacific. You can see up here, you see, we're going through the years very quickly as this globe rotates, but you're seeing seasonal changes in the subpolar regions of the Pacific Ocean. You can see seasonal changes in the Indian Ocean. Over here is going through monsoon and non-monsoon periods of the year. And the area that we were going to be operating, you can see some large changes here. So this is just to show that even, the variability you see in phytoplankton off your dock is probably buried within a much larger pattern, which is being driven by some pretty big processes. So a prominent phytoplankton group in the ocean are called coccolithophores. And these are important because these single cell phytoplankton produce scales that are made of calcium carbonate, otherwise known as limestone, or chalk. These are a critical part of the ocean's carbon cycle. And probably a quarter of all the marine sediments on Earth are made of this calcium carbonate that's produced by these single cell plants. And here's some pictures of these. They come in all sorts of shapes and sizes. I really don't have time to go through all of these, but this player right here is called Aemiliania huxleyi. It's ubiquitous around the planet, and it causes blooms. It makes blooms of this chalky suspension, and that chalky suspension makes the light very reflective, such that you can see these blooms from space, from satellites. And here's another really cool one. They have life cycles. One part of the life cycle will have large coccolith plates, and another part of the life cycle has little tiny plates on it. And to give you an idea of size here, this is a 5 micron scale bar, so 5 millionths of a meter. And just to give you an idea, you could fit 100 of these on the pointy end of a pin. So they're extremely small. And the White Cliffs of Dover in southern England are made of fossilized coccolithophores that have been accreting, sinking down and accreting over geological time. And you get an idea of size here. You know, there's a person standing up there on those cliffs. And indeed, over the entire Western European continent, it's covered with this deposit of chalk, which was produced by these coccolithophores. So, and this is obviously a big process. And the blooms of these things were actually discovered fairly recently in terms of science. These were discovered in 1983, these blooms, by a guy named Patrick Holligan here. This is a Nature paper he published. He later was to become one of the directors of Bigelow Laboratory. And he was working with space-based imagery. This is the first ocean color satellite that had been put into space. It's called the Coastal Zone Color Scanner. Charlie Yench, the first director of Bigelow Laboratory, was on the original NASA team that developed this instrument, the first ocean color sensor ever, and put it into space. And they saw these highly reflective features off of England and off of France. And so they said, let's get out in a boat and check it out. And when they got out there, they saw all of these are individual plates of Aemiliania huxleyi here. These guys had been eaten by a zooplankton, an insect of the ocean. This is actually looking at a close-up of a fecal pellet from one of these things. It's just packed with coccoliths. Here in the Gulf of Maine, we started looking at these in the late 1980s. And our first observations of a mesoscale coccolithophore bloom were made in the Gulf of Maine using a satellite called the Advanced Very High Resolution Radiometer. It's a big name, and it basically had a very crude camera on it that we could use to visualize high reflectance blooms. And we made the first observations of carbon fixation in coccolithophores in this feature. And here's what it looked like from space. So you can see Massachusetts right there in Nova Scotia. And this feature basically covered half of the Gulf of Maine. It was about 100,000 square kilometers. And we left from Booth Bay there was a very small research vessel. We called up the funding agency and said, look, we're looking at this thing. Could we get some money to go out there? And indeed, we went out, and when we got up the following morning, this is what the water looked like. Now, that's me in the picture. I had slightly darker hair then. And this was, I was wearing a navy blue jacket here. And this color, this is a very reflective bloom, and it was more like a turquoise color. This is not a great, this is taken from an old slide, but knowing that I was wearing a navy blue jacket, here's another picture, which is a little bit better color. That's me right there. There's Patrick Holligan and a postdoc at the time named Stephen Ackelson. And you can see how turquoise that water was relative to a navy blue jacket. You just see a little bit of navy blue there in my jacket. And if you filtered that water, this is what you would see. You'd be hard pressed to find a non-coccolith particle in that water. It's highly reflective, and you would have thought you would run aground in Bimini in the Caribbean. So just when we thought these blooms couldn't get any bigger, we saw this thing. And this feature was in the Northern Atlantic. The Grand Banks were over on this side. The Faroe Islands off of Scotland were over on this side. And here's Iceland. And you're looking at different shots from this AVHRR as waters were sort of mixing around there south of Iceland. And each of these, you can actually visualize eddies, big boluses of water here that contain these coccolithophores. That was in 1991 when we got into there. And that feature had a total area of 500,000 square kilometers. So imagine the whole North Atlantic lit up by these plants. And I had a colleague on the ship, Chuck Trees, who had he was my color chip for comparing color. Here he is up here. We're outside of the bloom, and you can see his navy blue sweatshirt. Here we are in the bloom, and again, you can see the turbidity of this water. If you put a light meter down into the water 2 feet, it would disappear. You couldn't see it. And it was like a sea of milk. That's pretty much what it looked like. So then when we thought we couldn't see anything bigger, we started putting together all the images into global views of the Earth. And especially in this region down here, we started seeing these high reflectance features in the Southern Ocean. At the time, we couldn't prove that these were coccolithophores. We had to actually go down there and take some trips across the Atlantic Basin and the Indian Basin to show that these were indeed coccolithophores. And we called this the Great Calcite Belt, which rings all the way around the Southern Ocean. 52 million square kilometers, 16% of the global ocean. And it contains about 1/3 of the chalk, or here it's called PIC, particularly inorganic carbon, but that's fancy words for suspended limestone or chalk from these microscopic plants. And the problem, people had ventured into this and published papers in peer-reviewed journals 14 times since the 1960s. And they described elevated concentrations here. There's another paper in the Indian Ocean over here, high concentrations and such. But it wasn't until with the advent of remote sensing, satellite remote sensing, that we had a vantage point that we could see that it indeed was one feature. And it was kind of, the early days was sort of like the blind people and the elephant, trying to describe what they were seeing, each of them going into an individual part of the belt. And then it's when we got back far enough, we could see that it was all due to one organism or a group of organisms. And it happens every year. Here's a collage of 10 years from December to March, basically the austral summer. And you can see this high reflectance water every year down in the Southern Ocean. This particular inorganic carbon, again, is just chalk. So the lighter blue areas are the higher concentrations. So we then wrote a paper, and this was, we were writing it in 2015, about the same time that we had the idea for the work, this latest trip, to go down there and make measurements to see what was there. And we basically showed that in all these areas, as we moved along, the elevated reflectance was due to suspended calcium carbonate. And this went from South America, South Africa, all the way over to Western Australia. And here's a scanning electron micrograph of another type of species called Discosphaera tubifera. And that central area there is about 5 millionths of a meter. Again, they're just spectacular. So we're going to take a little sidebar here for a moment and talk just for a second about Bigelow's Art Science Collaboration Program. And I'm introducing you here to Julie Crane, who comes from Camden, Maine. She's a multimedia artist who approached Bigelow, and she has interests in the question of how we are impacted by the influencers that we can't see, like the microscopic plankton. And so she came down, she talked with all the members of the lab group. We showed her how we measure coccolithophores and their optical properties and their biogeochemical properties and how they relate to the carbon cycle. And then she's been involved in a project to make a giant-sized Emiliania huxleyi, which she has made great strides. Here she's shown on the left with these things on the beach. And then on the right, you can see she's actually made the sphere that she's going to mount these things on. And this will be on display, if I understand correctly, at Bigelow Laboratory, not too far from now. Okay, that's a little sidebar. Now, I have to tell you a little bit about a water mass. And you say, a water mass, isn't water all the same around the planet? And the answer is no. There are different types of waters that have different temperatures and different salinities, and we use those properties to identify them. And this water mass I'm going to tell you about is subantarctic mode water. Subantarctic mode water. And it's a water mass that forms in the Southern Ocean. And it's formed in the winter months, and it winds up getting transported to the north underwater. It's formed in the south, in the Southern Ocean, and then it dips down and heads to the north. And this water is estimated to control 3/4 of the biological production of waters north of 30 degrees south parallel. And I can just hear people saying, where the heck is the 30 degrees south parallel? So I have this little, this little fly-in here, which shows the globe. And here's the 30 degrees south parallel. And this guy, Sarmiento, wrote this paper in which he was saying that 3/4 of the biological productivity of the ocean is being controlled, and that's north of 30 degrees, is being controlled by this water mass. And so how does that work? Diatoms, which is a type of phytoplankton that make little glass houses made of silicon, they are growing down here in the Southern Ocean. and they're growing in the subantarctic mode water. And they're, as they grow, they take up the silicate, the silicate, the nutrient, and before that subantarctic mode water moves to the north, they basically scavenge it out. So none of the water heading to the north necessarily will have a lot of silicate. Or if they don't grow a lot down here, then silicate will leak to the north, and that silicate will then control the productivity of diatoms in the North Atlantic. So diatoms, in essence, are conditioning the water in the Southern Ocean before its long trip to the north. And the thing I need to point out to is, you see my cursor here. This is in the area of the formation of subantarctic mode water, and it takes about 40 years, 40 years, to make it up to the equator. So that may seem like a long time, but in the time scale that ocean currents move in the deep sea, that's actually pretty fast. So, and just to drive home the idea about conditioning, there's an analogy, an air conditioner, which basically changes the temperature and the humidity of the air. Or you could have a hair conditioner that changes the chemistry of your hair follicle, and it can add oils back and things like that. And algal growth here changes the chemistry of the water by taking up certain nutrients, which later on, when that water resurfaces further to the north, may or may not be available for algae to use in the Northern Hemisphere. So the question that we were asking was whether the conditioning could affect what will grow further to the north. But we were now going to focus not on diatoms, but on these other plants called coccolithophores. So I have a diagram here, kind of a three-dimensional diagram. The continent of Antarctica is here. Here's South America. There's South Africa. Here's Australia and New Zealand. And this basically shows you these different water masses around the Southern Ocean. And these water masses are separated by boundaries. They're like frontal boundaries on a weather map, where you'll have water of one temperature and salinity on one side and water of another temperature and salinity on the other side. Well, what happens is the Great Calcite Belt sits just north of the diatoms. So what does that look like? So here is where the diatoms form in the Southern Ocean. This is what Sarmiento was describing, and he added this subduction or sinking flow here to the north, and the diatoms sit right at the top here, and they scrub out the nutrients before the water heads to the north. So they do their some conditioning. The Great Calcite Belt basically sits north of the diatoms, and it also is in the subantarctic mode water, flows to the north, goes down deep to maybe 700 meters deep, or even 1,000 meters, but that's not as deep as the ocean. And the coccolithophores sit right at the surface, and the question was, how are they conditioning the water, which would be different from how the diatoms are conditioning the water. And there's one little fly-in I need to point out here. And in this particular figure, the Southern Ocean waters are warming at 0.02 degrees Celsius per decade. That may not seem like very much, but that's actually quite a bit. That's a degree every decade. I'm sorry, a degree every 5 years. And this is just to show, you know, as we warm the planet further through introduction of fossil fuel and CO2 into the atmosphere and the greenhouse effect, we warm this area. That will tend to cap off, to cap off this surface layer, which will slow the flow of those nutrients. So the concern is, and we have to be aware of what we can do to try to keep the planet from getting more CO2 into the atmosphere, to try to prevent things like this, of capping this, because this is incredibly important to the productivity further to the north. Okay, in the last couple of minutes, I'm gonna show you some preliminary results. So this is the cruise track. You've seen it before. We came in here and we looked at a region called the Agulhas Meander. Right here, there's a big current that comes down and does what's called the Agulhas retroflexion, and then it meanders in this region. And I've been looking at this for 20 years. And when the captain said we couldn't go further down to the Crozet Islands, say, and get down here where we proposed to go, I said, let's go here. And we've been watching coccolithophores in this area for years. Let's study what's making them grow here. And then there happened to be an eddy, just like the eddy that I showed you earlier south of Iceland. There's an eddy spinning around up here. And I said, we'll go visit that. We'll survey it. We'll dodge into the shipyard, come back down, study this feature again, come back up, study the same eddy again, and then we'll hightail it for Mauritius. So how do we know where these eddies are? We need to understand something about the physical oceanography. And we sampled these eddies that basically had trapped water with coccolithophores in them. So imagine you could look down from space and you could see 100-kilometer, 60-mile wide bolus of water lumbering around in a clockwise fashion that was like a gigantic culture vessel, a gigantic flask where you had these coccolithophores growing. And what you're looking at here is called satellite altimetry. And I don't have a lot of time to describe this except to say the ocean is not level. As much as you may think it's level, yes, there are waves and such, but currents make for little mountains and little troughs in the ocean. There could be a difference in elevation of 1/2 meter between here and here. And so the altimeters use radar to measure the distance or the height of these, or the depth of these, the sea surface feature. And we could tell that there was a current going around like this. And this was sort of, it was a filament. It's like a stretch of water that stretches to the north. And it happened to be full of coccolithophores. And on about December 25th, and this eddy here spun off from the top of that filament and was lumbering around out here. So we actually had a time point at which this eddy was born, and we could look at how the plants were conditioning that water as that eddy spun around. And here's a view of the altimetry from February 8th, and here's a view from February 22nd, same features, and we were just looking at how things were changing. And basically in the eddy here, it was like an entrapped culture, and things changed. So here's a picture of the video plankton recorder. We're getting ready to deploy it. The ship is brought around onto its new course. And this thing, and I have a close-up of it, everyone's ready with their taglines, and the A-frame is up. And now I'm going to zoom in on that. And this thing is packed with gear. Some of our optical gear was on it. There's strobes here, and there's camera here that are basically looking at all the particles that go whizzing by. It basically undulates through the water column down to about 100 meters depth as we steam along at about 8 knots. And this allowed us to survey features and get data like this for temperature and salinity, dissolved oxygen, light scatter, and basically build a 3-dimensional picture of what these eddies look like. And through this helped us to define the factors that affect the growth of the coccolithophores. This is a simple, I don't show a lot of data here, but this is a simple temperature versus salinity plot. There's fresh water on this end of the scale. There's salty water at this end of the scale. There's cold water down here, warm water up here. And the color here tells you when there are a lot of coccolithophores, if it's red, or if there are no coccolithophores when it's blue. And the density of the seawater are these gray lines. So the dense water is down here, and the less dense water is here. This is how water masses move around in the ocean. Less dense tends to be on top of more dense. And all the take-home message here is that the coccolithophores love this water from the subantarctic frontal region. It was a coccolithophore party here, but when you got to other regions up here, like when we were in this part of the transect, we found way fewer of them. What did the water look like? Here is Julia Middleton, who's a former Bigelow Laboratory Colby alumni. She's now a doctoral student at the Woods Hole Oceanographic Institute. And I had her come over with her navy blue Colby sweatshirt to pose here in front of the water so you could get a feeling for the color difference from the navy blue, more typical ocean blue. You could see the higher reflectance here. But to really get an idea of what the reflectance looks like, you have to go aloft. This is from the main bridge wing, looking down on the water. And you could see it was much greener than it appears here, and it's much more turbid. This is what the Great Calcite Belt looks like, close up. We did deck experiments on board in which we had to control, or we added different nutrients. And there's that iron I mentioned earlier on, and we add combinations of nutrients. And we also added subantarctic mode water to see if it caused any effect. And only the nitrate and subantarctic mode water had any effect on the algal growth. We called this the secret sauce. And there was no effect of iron, which was a real surprise. In this part of the world ocean, typically iron is limiting, and there is no effect of silicate that we could see. Here is a description of some results by my REU, Research Experience for Undergraduate Student, Hannah Primiano. This year, our REU program with COVID-19, basically we couldn't have an in-person REU program, but we decided to do it virtually. And Hannah was working on microscope images that we had taken from the cruise, where we could see all the algae, we could see just the coccolithophores, we could see all the fluorescent phytoplankton that had chlorophyll in them, or the cyanobacteria. And Hannah has shown that it isn't just the coccolithophores that were responding to enhanced nutrients. Dinoflagellates were responding as well. They were covariant with the coccolithophores. And cyanobacteria also covaried with the coccolithophores. So they all appear to be responding to the same factor. And some of the greenness in that photo that I showed you earlier is the fact that there are other algae in there besides the coccolithophores. So here's the old timeline. And we're going to add on to this a little. There was the cruise coming back to COVID. And here's the, we found out we were supposed to be going to sea in January 21. We found out cruise 2A had been canceled because of COVID. We were going to be in the Indian Ocean, but then we've now actually started planning. We're going to be looking at subantarctic mode water in the Southwest Pacific. And that's near American Samoa. And we'll be on the research vessel, Scripps research vessel, Roger Revelle. And here's a picture of Roger Revelle. It's also an Agor 24, just like the Thomas Thompson. And if you look really carefully in this picture, you'll see that there's a rainbow on the right side, which I take as a particularly good omen. So in closing, what can you do to support ocean science? Support organizations that do basic research. We're not the only ones out there that do it. We certainly would appreciate your support. Communicate with your elected representatives the importance of basic research in earth science. Learn about how man is changing our planet and change your behavior. Be an agent of change. And I would add that during this pandemic, we've had it's a terrible thing, but it's been a rather unique opportunity when everyone stopped driving and everyone stopped flying, that the trajectory of CO2 on the planet, the rate at which it's increasing, has actually dropped measurably, statistically significantly. And that just shows that, you know, if we have to get off of carbon as a fuel source for oil, that we could actually affect the trajectory of CO2 and climate change on the planet. And finally, I know it's probably a little late. You've got a couple hours to get to the polls, but vote. And So I'd like to thank the National Science Foundation, and that's really taxpayer supported. That's you. Thank you so much for funding this basic research. My Bigelow Lab group here, who know what I'm thinking before I even think it, and these guys are just fantastic. And the science party of Thomas Thompson TN 376 shown here, all 26 or 28 of them. And some of them might have been asleep for that picture, but anyhow, thank you very much. And I'll take questions and use the Q&A button at the bottom of your screen to submit the questions. Thank you.
Great, thanks, Barney. Great talk. First question we've got here is about the effect of atmospheric CO2 on these organisms. Do they trap some of that CO2 as their chalky coccoliths that they create? Yeah. And if so, have we seen an increase in them as a result of atmospheric CO2 increases?
Wow, that's a big question. Okay. First of all, coccolithophores are unique amongst the phytoplankton on short time scales. So if you put these things in a flask, or even if you go into an eddy that's loaded with coccolithophores, you will see the concentration of CO2 increase over time. The process of calcifying produces, you take two bicarbonates and you make one CO2 and one calcium carbonate that goes into the coccoliths. Now, the plants will use some of that CO2 in photosynthesis, and so they'll take that CO2 right up, but they don't always take up that CO2. So on the short time scale, you'll actually see the CO2 go up by a factor, it's in science terms, it's about 40 parts per million. In the long time scale, so over years, this calcium carbonate that's been formed, it's a rock, it sinks, and it carries with it a lot of other organic matter, which is called the biological pump. And that dense material takes with it a lot of carbon to the bottom of the sea, and it scrubs it from the surface ocean. So phytoplankton in general they're growing and decaying in the surface ocean, but it's the coccoliths that provide significant ballast to get all this stuff down deep. And if it weren't for organisms like coccolithophores that weigh down this organic matter, the partial pressure of CO2 in the atmosphere would be way higher than it is now. So they do have an impact. Let's see, the second part of that question, Stephen?
Was just, have we seen an increase in those organisms as CO2 in the atmosphere has increased?
Yeah, okay. So there was a paper published in Science a few years ago looking at a large data set that is just for the North Atlantic. Organisms like the coccolithophores were actually changing, were going up, as it turned out. And other organisms like the diatoms appeared to be going down. So there are some changes happening in places like the North Atlantic Ocean. Is ocean acidification impacting them? It's a little hard to say because the coccolithophores are made of calcite. It's a different form of the mineral than say a mussel would be. That is a mussel in the ocean or a clam. And that's a little harder to dissolve. So, but the acidification makes it harder for these organisms to calcify. It costs them energy because they have to work harder to calcify. So generally it's thought that it has a negative impact, but there's a counter effect going on here, which is that as the surface ocean warms, it's stratifying it more, and it's holding the nutrients down deep in the ocean, so they can't punch into the surface ocean. And that means, though, that's the type of environment that a coccolithophore absolutely performs incredibly well in. It can outcompete other algae in waters that are moderately productive, that are stratified, where a diatom would just pack it up. It needs higher nutrients, it needs more mixing. The coccolithophores do well in warming stratified water. So I think that's why we're seeing the increase in the coccolithophores.
Thanks, Barney. Since your group went to the Indian Ocean side of the Southern Ocean in your first cruise, will your experiments be affected now that you'll be sailing on the Pacific side?
Good question. Actually, no. So as I mentioned, that there's a slide, that sort of three-dimensional slide with Antarctica and the circulation, the Southern Ocean circulation going around it and the subduction. Subantarctic mode water, one of the fortunate things about why we were able to move the cruise is subantarctic mode water is formed in every ocean basin. It's formed in the Atlantic, in the Pacific, and in the Indian. And so it was actually relatively easy for us to move, and we'll do the same experiments, and we'll use the same remote sensing of that part of the world ocean to navigate ourselves to features that are like these eddies, for example, that have high concentrations of coccolithophores in them. So we lucked out on that one.
Great. We've got lots more questions, but we're short on time. So we'll just answer one more here.
Okay.
Someone asked about how long do you think that scientists will be working with the data that you collect on these cruises?
Well, as people will be working on this data for decades, probably, because as the National Science Foundation requires of us to deposit our data in a public repository, which basically gives anybody and everybody free access. And so other scientists, we will be putting our data in there. And there's a lot of other data that I didn't even talk about that the ship is constantly collecting because it's so expensive to get out there and make these measurements. So everything from bathymetry and meteorology and just water chemistry and physical properties of water, that all of that data goes into this database. We ourselves are writing papers and processing. We're actually processing samples now still, and we will be processing samples for some time. After we process those samples, we will publish peer-reviewed papers on all of this. And then other people, they start picking up on the data sets. And the data will be used well into the future.